Good afternoon students, welcome back to botany session, and in these videos we are going to discuss the cell wall of cell biology. It is one of the 11th chapter cell biology. The topic is cell wall. So what is called cell wall, how it is formed, what are the types of cell wall, these are the topic now we are going to discuss. Cell wall, it is one of the outermost and protective covering of the cell. These are called cell wall. Outer border that give the shape to the cells. It is a protective covering of the cells. These are called cell wall. The cell wall generally present in bacteria, blue-green algae. It is also called cyanobacteria. Then fungi, then plants. Generally, it is absent in animal cell. So, cell wall present in plant cell only. In addition to that, bacteria, fungi, it is absent in animal cells. So, the cell wall is first observed by Robert Hooke. He is the first person have been reported cell wall. He is also first person to reported cell. How he identified cells? From the thin cork wood, he observed a honeycomb-like structures, and each boxes or box-like structures he is called as a cell by Robert Hooke in the year 1665. And he is also observed the outer covering of the cell after the inventions of electron microscope. After the invention of electron microscope, each box is covered by outer covering is called cell wall. It is observed by Robert Hooke. But the cell wall of these various organisms is composed of different compositions in various organisms. What are the different complex substances are present in the cell wall? In bacterial cell, the cell wall is made up of peptidoglycans. It is nothing but peptide, that's a protein. Glycan, it is a sugar. So, the bacterial cell wall is made up of peptidoglycan, it is nothing but protein and sugar. Then blue-green algae, it is also called cyanobacteria. The cell wall is made up of BAPA, I mean pimeric acids. Sorry, DAPA, diaminopimelic acid. Diaminopimelic acid. Blue-green algae are also cyanobacteria. Fungi, the cell wall is made up of chitin and fungal cellulose. Then plant cell wall is made up of cellulose, hemicellulose, pectic substance. In addition to that, cutin, suberin, lignin. In addition to that, silica also deposited. So the plant cell wall is made up of cellulose, its a primary substance is cellulose. In addition to that, hemicellulose, pectic substance, cutin, these are the fatty substance, cutin. Suberin, it is also fatty substance. In addition to that, lignin and silica. Then algal cell wall. Algae cell wall is made up of primarily cellulose. In addition to that, galactans, then monans. It is nothing but polysaccharides. Monans is nothing but polysaccharide. So algae, it is based on the pigmentation. There are three types of algae: red algae, brown algae, green algae. Then in addition to that, blue-green algae. Algae, it is also the manufacture carbohydrate due to the presence of a green color pigment is called chlorophyll present. But here also cell wall is made of, apart from the plant cell, apart from galactons is present, then mannans also present. But cellulose is a primary component, both plant cells, then chitin, fungal cell wall, it is also composed of fungal cellulose, plant cell also composed of cellulose. In addition to that, galactons and mannans also present. These are the various chemical compositions are present in the cell wall of the various organisms. The next, in plants, the cell walls that shows three distinct regions. It shows three distinct regions. One is primary wall. Second, secondary wall. Third, middle lamellae. Then fourth one is tertiary wall. So these are the various regions, distinct layer is present in the cell walls. I will explain one by one. If you take first one is a middle lamellae. It's a better to understand. It is a cell. See, it is a middle lamellae. Then what is called middle lamellae? It is a thin amorphous, a cementing layer that join the adjacent cells. The two adjacent cells are joined by the cementing and amorphous. Amorphous. It is nothing but a shapeless layer is called middle lamellae. It forms a bridge between the two cells. See, it is another cell, neighboring cell. It is another cell, it is another cell. The two cells it joined by a layer, these are called middle lamellae. The middle lamellae, it is composed of calcium pectate and magnesium pectate. Calcium and magnesium pectate. Where it is formed, how it is formed, this middle lamellae. Middle lamellae is formed during at the time of cytokinesis. Cytokinesis is nothing but division of cytoplasm. A cell, it is divided into two cells after the completion of two processes. One is karyokinesis, another one is cytokinesis. Karyokinesis is nothing but division of nucleus. It takes place 4 stages. Metaphase, anaphase, prophase, metaphase, anaphase, telophase. After the telophase, nucleus is completely divided into 2. Then followed by cytokinesis, nothing but division of cytoplasm. After cytoplasm are divided, the two cells is formed. Between the two cells, there is a cementing layer, amorphous layer is formed, is nothing but middle lamellae. It is made up of calcium pectate and magnesium pectate. Now, it's a questions. Retting of fibers. Retting of fiber. Then softening of fruit. Softening of fruit. You know what's called retting of fibers? The process of separation of fiber is called retting of fibers. Fiber is obtained from cell wall of fiber yielding plant. For example, jute and hemp. Sunal, jute and hemp. It is obtained from cell wall. How the fibers is easily separated from the cell wall? By the process called retting. If the fibers are plants are kept under the waters for few days due to the presence of a saprophytic bacteria, they act on the cell wall of plant cell, which help to decomposition of the cell wall. As a cell, fibers are easily separated. So, question: which portions of the cells are gets decomposed? Middle lamellae. Especially this calcium pectate and magnesium pectate of the middle lamella gets decomposed due to the presence of an enzyme is called pectic enzymes. The pectic enzymes are released by the bacteria. Apart from that, softening of the fruits. Fruits may get softening at the time of ripening. Fruit ripening, they become soft due to the dissolutions of these calcium pectate and magnesium pectates. So, it is thin amorphous that forms a bridge between the two adjacent cells is called middle lamellae. So, first middle lamellae open. The next is primary wall. What is called primary wall? It is a thin membrane and the extensible membrane lies inner to the middle lamellae. These are called primary wall. It is primary wall. Actually, primary wall, it is the outermost layer of the cell. It is a cells. Cell is nothing but a protoplasm. Protoplasm is a center portion, that is a living component of the cell is protoplasm. Protoplasm is covered by cell wall. So, cell wall especially three layers: outer middle lamellae, inner to the middle lamellae primary wall, inner to the primary wall secondary wall. Then next protoplasm, center nucleus is present. So, primary wall, it is a outermost cell but lies inner to the middle lamellae is called primary wall. The primary wall primarily composed of cellulosic microtubule. Primary wall, it is filled with a jelly-like matrix substance called pectic substance. But primarily it is composed of cellulosic microfibril. This is a primary wall. It is filled with a filling material is called pectic substance, pectin. In addition to that, these are a network arrangement of microtubules. It is a microtubules. How they are oriented? In different angle they gets oriented. They are arranged in the network like such as called microtubules. But most of the plant cell, the microtubule, it is made up of cellulose. There are many cellulose is present. Cellulose, it is a polysaccharide. It is made up of many monomer, glucose. See, it is a microtubule, microfibril. Microfibril is made up of cellulose. You know that cellulose is a 6-carbon atom. Cellulose, first carbon, second carbon, third, fourth carbon. So, it is a glucose. Many monomers are linked together to form polymer. So, one glucose of the first carbon, another glucose of the fourth carbons are linked together by 1,4 glycosidic bond. But due to the presence of 1,4 glycosidic bonds are present, they are linked together to form a polymer. So, most of the plant cell, this microfibril is made up of cellulose. The next, the cellulose are which is present in the matrix substance. Primary one, matrix substance is nothing but made up of pectin. Yes, it is a pectic substance. In addition to the pectin, it is the matrix also contain. It is also contain hemicellulose. Hemicellulose, pectin, glycoprotein, glycoprotein plus water also present. Remember, primary wall lies inner to the middle lamella. It is filled with a filling substance is called matrix. Matrix are filled with the pectic substance. Then in the matrix there is a differently oriented network structure is called cellulosic microfiber is present. These are the primary component of the primary cell wall. Cellulosic microfibril. The cellulosic microfibril is made up of cellulose. Then what are the functions of this hemicellulose, pectin, glycoprotein present in the matrix? What are the functions of this hemicellulose? This hemicellulose, yes, it is a hemicellulose which bind the microfibril to the matrix, fixed together due to the presence of hemicellulose. What are the functions of this glycoprotein? Yes, it is a glycoprotein. The glycoprotein which help to orientations of the microfibril. I mean different angles. They are how they are oriented in different angles. The orientations of the microfibril is regulated by glycoproteins. What are the functions of this pectic substance? These are the filling material which is present in the primary cell walls. It is in addition to that where it is present. For example, meristematic cell and parenchyma. Parenchyma and meristematic cells. Parenchyma, you know that it is one of the simple tissue. Parenchyma, collenchyma, sclerenchyma. In the parenchyma cell, only primary wall is present, secondary wall is absent. In addition to that, meristematic tissue, where is called meristem, it is actually dividing portions of the plant body. It is present the root tip, shoot tip, that is a meristematic region. In the meristematic cells, have only primary wall, here secondary wall is absent. So primary wall over. Next, secondary wall. The secondary wall lies inner to the primary. It is differentiated or divided into three sublayers. It is a S1, I mean secondary wall 1. It is S1. In addition to that, S2. It is S2. This S2. Then S3. This S3. These three layers are constituted to form secondary wall. So the secondary wall, it is a thick, inelastic wall. Primary wall, it is a elastic, thin, elastic, extensible wall. It is secondary wall lies inner to the primary wall. It is a thick wall, inelastic wall. It is formed during at the time of cell maturation. At the time of cell maturation only the secondary wall is formed. It is not present in all the cells, except to especially only during the time of maturations only. It is primarily composed of, secondary wall primarily composed of cellulose, hemicellulose, then pectic substance. Pectic substance is present. But the growth takes place. Yes, of course it is a the thick cell formed during at the time of maturation. After maturation, maximum growth of the cells are get arrested, even though the secondary wall growth takes place by accretion. Accretion. What do you mean by accretion? I mean deposition of material within the existing cells, within the existing surface of the cell structure. They are already there. Certain materials are already there on the surface of the secondary wall. In addition to them, certain secondary wall substances also get deposited as a result increase in the thickness. So what are the deposition material? For example, QT, suberin, lignin. So these are gets deposited on the secondary wall as a cell growth takes place. Cutin, where is present cutin? Cutin is present in the outermost layer of the epidermis, especially the stem, both monocots and dicots stem. Outermost layer of the stem is called epidermis. Epidermis are covered with the cuticles. But the function of these cuticles, it is highly impermeable to prevent the water loss, to check the loss of transpirations. Cuticle. Suberin, both the cuticle suberin, these are the fatty substance. Suberin is present in the inner and radial tangential wall of the root. It is present only in the root. Root endodermal cell. Endodermis, there are two layers are present. I mean two wall. One is a tangential wall, another one is a radial wall. It is a tangential wall, it is a radial wall. So the suberin is deposited only on the radial and the tangential wall of the endodermal cells. It is also highly impermeable. It prevents the entry and exit of waters. So suberin. Lignin is present in the tracheids of gymnosperms and vessels in the angiosperm. So in gymnosperm, which is a water connecting elements, tracheids, they have lignin deposited. Secondary wall is deposited with lignin. Similarly, vessels are the chief water connecting elements in angiosperm flowering plants. Lignins are deposited in the vessels also. So the process of deposition of lignin is called lignification, suberification, cutinations. So these are present in the secondary wall. Then in addition to that, it is also composed of this microtubules. Microfibrils, cellulosic microfibrils are present both in the primary wall as well as secondary wall also. But what are the different? In primary wall, they are loosely arranged. The microfibrils are loosely arranged in a network like such as. But in the secondary wall, they are tightly packed in the different orientation. So it is a orientation. It is a orientation. In different orientation to form a thick sheet, thick lamination sheet. It look like a thick lamination sheet due to the presence of these microfibrils. As a result, increase the thickening of the secondary wall because of this cellulosic microfibril. Different cellulosic microfibril in the primary wall is loosely arranged. In the secondary wall is tightly packed. These are secondary wall is over. The next tertiary wall, it is a rare. Inner to the secondary wall, tertiary wall is present. Central lumen of the cells. I mean, it is a protoplasm that composed of both living and non-living substances are present. Center nucleus cytoplasm is present. So these are the maximum three distinct layers are present in the cell wall. First one is actually middle lamellae. Second primary wall. Then third secondary wall is present. Next, plasmodesmata and pits. Then pit. Plasmodesmata. It is act as a channel between the protoplasm of the two adjacent cells. See, this is a middle lamellae. This is a primary wall. Yes, These are the two neighboring cells, center, middle lamellae, primary wall. Both the neighboring cells are connected through plasmodesmata. This plasmodesmata. So the plasmodesmata that act as a channel way between the protoplasm. Yes, it's a protoplasm of the one cell. It's a protoplasm of the neighboring cell. These two protoplasm of the neighboring cells are connected by plasmodesmata. It is a small pit, so through which many substances are passed through. Both the harmful substances, living substances, non-living substances, from one cell to the another cells, they are easily diffused based on their osmotic pressure from higher concentration to low concentration, from low concentration to higher concentration, either by active process or passive process, takes place through a bridge is called plasmodesmata. So sometimes this endoplasmic reticulum, yes, it is a nucleus. Nucleus is surrounded with the endoplasmic reticulum. The endoplasmic reticulum which is passed through the plasmodesmata, these are called Dysmore tubules. Tubules. Then what do you mean by Dysmore tubules? They are extensions of endoplasmic reticulum. It is passed through the plasmodesmata into the neighboring cells. These tubules is called Dysmore tubules. So plasmodesmata over. The next pit. Then what do you mean by pit? Similarly, yes, it is a middle lamellae. It is a primary wall. Pit. So, the secondary wall, middle lamellae, it is a continuous layer, evenly thickening. Primary wall, it is also evenly thickening. Moreover, at a few region, the secondary wall is unevenly thickening. Look, this is a secondary wall. Already I point out secondary wall actually is subdivided into three layers, S1, S2, S3. The secondary wall here it is unevenly thickening. They left some space. These places are called pit. But whereas in middle lamellae, then primary wall is continuously or evenly thickening. Secondary wall is unevenly thickening at some region. Such regions are called pit. There are two types of pit. It is a secondary wall. The pit actually there are two types of pit. One is a simple pit, another one is a bordered pit. Yes, it is a simple pit. Another one is a bordered pit. So what is called simple pit and bordered pit? The simple pit have same width at the both the side. The width have the same width both the sides. These are called simple pit. But whereas in bordered pit, the secondary wall is hang over on the both sides. Middle lamellae, primary wall, primary wall, it is a secondary wall. Secondary wall. So the secondary wall which hang over on the both sides, these are called bordered pit. Both pits, simple pits and bordered pits are the neighboring cells which is lies in opposite to each other. It's a what other cell? It is opposite cell. In both the cells have the pit. The pit is lies in opposite side. Pit is lies in opposite sides. These are called pit cavity or pit pore. It is a pit chamber. Pit chamber. These three are pit membrane. Pit membrane. So once again, pit, there are two types of pit. One is a simple pit, another one is a bordered pit. Simple pit have the same width on the both sides. Bordered pit, the second wall is hang over. To form a pit is called bordered pit. Both the pit, simple pit and the bordered pits, they have pit chamber and pit membrane. It is a pit membrane. Pit membrane. It is a pit chamber. Pit chamber. In bordered pit, the pit membrane have a minute pore which is made up of a fatty substance is called suberin. Such minute pore is called torus. Then what are the functions of this torus? It regulates diffusions. You know what is called diffusion? Movement of any gases or liquid substances from its higher concentration to its lower concentration through a minute pore is called torus. Especially in the bordered pit, this diffusion is regulated by a minute opening is called torus. So neat question. Torus is made up of a fatty substance is called suberin. Once again, in bordered pit, the pit membrane has a minute pore which is made up of a fatty substance called suberin. Such minute pore is called torus that regulate diffusions. So pit over. How it is pit is formed? Due to the uneven thickening of the secondary walls. Two types of pit. Then next, what are the functions? What are the functions of this cell wall? The first functions of cell wall that give the definite shape and rigid to the cells. Definite shape and rigid to the cell. Wow. Cell wall, it's only bordering or outer covering, protective covering of the cells. It's not a cell. Protoplast actually is a cell. It's a living part of the cell. That's nothing but protoplast. Protoplast is well protected by plasma membrane, then cell wall. Cell wall actually it is a three layer, middle layer, primary wall, secondary wall. Some cases are tertiary also present. So it gives the definite shape to the cell due to the presence of this cell wall and also rigid to the cell because of cell wall. The second one is it serves as a barrier. Barrier for many molecules which is enter into the cells. See, a lot of harmful substances or many chemical substances also may be entered entered into the cells and exit of the cells. It is takes place through a minute opening is called plasmodesmata, and also torus also may be carried out. See, it is a barrier. The cell wall acts as a barrier for the many molecules. How? Primary wall actually it is a permeable membrane. They are allowed to pass through all the substance without any barrier. Primary wall. But where is the barrier? Secondary wall is a barrier because it is made up of certain secondary wall substance, lignin, suberin, cutin. These are highly impermeable. It does not allow to pass through it. If suppose the secondary wall is allowed to pass through all the substance, certain harmful substance may be entered. They cause destructions of the cells. So to prevent the entering of this harmful substance, secondary wall is a barrier. That is the advantages of the cell because of barrier. So once again, this cell wall that is act as a or serves as a barrier for the several chemical substances enter into the cell. The third one is it provides protection to the inner protoplasm from mechanical injury. See, protoplasm is a center. Yes, it is a protoplasm center. The protoplasm, inner protoplasm, it is highly protected from the mechanical injury due to the presence of outer covering is called cell wall. So it provides protection. Protections of protoplasm from mechanical injury. The next, to maintain the turgidity of the cells, I mean to prevent bursting of the cells. Prevent bursting. Another functions of cell wall, it prevents bursting of the cell and also to maintain osmotic pressure by the maintenance of osmotic pressure. How to prevent the bursting of the cells? Usually, water molecules move from low osmotic pressure to the high osmotic pressure, and the high osmotic potential to the low osmotic potential. These are the wave movement of water molecules. If take in the leaf, I already pointed out in previous videos, if the water molecules are high, enter into the cell through rooters, they increase the volume of the cells. Why the water molecules are enter? Inside the root there is a high osmotic pressure. Outside the root it is a low osmotic pressure. So movement of water molecule from low osmotic pressure to the high osmotic pressure. Apart from that, high osmotic potential to the low osmotic potential. How the inside the plant cells to create more osmotic pressure due to the presence of solution. Leaf, it is an important photosynthetic organ. They synthesize carbohydrate if they are mingled with water to form a solution. If the solutions are gets increasing, osmotic pressure is increasing. That is why water is automatically enter the cell. If the water is continuously enter the cells, they increase the volume of the cells, and in some cases it may be burst. So yes, under this situation, the cell is said to be turgid. The cell is turgid to maintain the shape of the cell. Similarly, if the cell is placed under the hypertonic solution, salt solution, sugar solution, exosmosis maybe takes place, the cell's protoplasm becomes shrinkage. So, to prevent the burst of the cells due to the presence of cell wall, if the water is continuously enter, increase the volume, cell wall is a barrier, cell wall is outer covering to prevent the bursting of the cells. It's another function. And to maintain osmotic pressure also. The last one is, it is a defense mechanisms. Defense mechanisms. Cell wall is play an important role by act as a defense mechanism. How it is act as a defense mechanism? It protects the cell from external environmental factors, light, temperature, humidity. Prevent these external or environmental factors. It is act as a defense mechanism. These are the functions of cell wall. Thank you, students. Next videos we will discuss next class.